In Chapter 7, we're going to look at cell reproduction. Remember mitosis, or how cells actually duplicate themselves. So, the objectives today are to actually look at the role of mitosis and meiosis in the normal life of a cell. We're going to differentiate between chromosomes and genes, autosomes and sex chromosomes, diploid and haploid cells. We'll go over the cell cycle, which is basically a day in the life of a cell, look at each phase of mitosis and the interphase between the mitotic phases. And we'll look at the significant events in each phase. So we want to go kind of all the way through the cell cycle and look at how mitosis is completed in these phases. We'll also look at cytokinesis. And understanding chromosomes, we want to look at all of the chromosomes together in something we call a karyotype and learn how to use that to diagnose certain diseases. So then, after the cells duplicate themselves, we also want to learn how meiosis creates haploid cells. In other words, cells with half of the genetic information. This would be our gametes, sperm and egg. And during the process of meiosis, how we get opportunities for genetic diversity through events of crossing over and independent assortment of chromosomes. So we'll also look at how problems that can occur during meiosis, such as non-disjunction, can give us the wrong number of chromosomes. These are things like trisomy 21, which is Down syndrome, or if we're missing an X chromosome, that's going to be Turner's. If we have an extra X chromosome in a male, that's Klinefelter's. So abnormal number of chromosomes can cause specific genetic phenotypes. So let's start with looking at the two types of cell division. So remember, you began life as a single cell. So when sperm met egg, we call that a zygote. That's a crazy name, right? The zygote is going to have all of the DNA from the mother in the egg and from the father in the sperm. We call this a diploid or 2N number. And in humans, that would mean 46 chromosomes in that single cell. So how did that happen? How did we create a sperm and an egg? That happens through the process of meiosis, where we basically have our full complement of chromosomes, so 46 chromosomes, being split in half to form a haploid number, or N. In case of humans, this would be 23 chromosomes, and that'll be in our gametes, our sperm and egg. Once we have a fertilization occur and we have a zygote growing, that initial single cell is going to duplicate itself through the process of mitosis. And here we're going to get identical cells all throughout your body. They are going to differentiate into different tissues like your skin, your muscles, your bone, but they're all going to have the same genetic information. Throughout your body, we call all of these cells somatic cells. These have a diploid number, so they have 46 chromosomes. And this process is used for original growth and also repair of tissues. So this is kind of just looking at the life cycle of an organism, particularly a human, and how these two types of cell divisions interact. So here we have mature adults, a male and a female. Throughout their body, their somatic cells would be diploid, or 2N, carrying the 46 chromosomes. At puberty, in the female, in the ovaries, the process of meiosis will occur, producing the haploid egg. Similarly, in the male, in the testes, meiosis will produce the haploid sperm. These are the gametes, which can fuse to then form the zygote and the next generation, which will again be diploid. So we just have this continuing throughout the life cycle. So this is what we just talked about. In meiosis, we produce gametes that have half the number of chromosomes. In females, this occurs in the ovaries, producing eggs, and in males, it occurs in the testes, producing sperm. Again, looking at the overall life cycle. So let's talk first about those chromosomes. I just said that in a normal human, you would have 46 chromosomes. 23 of those came from your mom, and 23 of those came from your father. So what is a chromosome? This is the genetic material that's in the nucleus, and it is very tightly coiled. So remember, we talked about that DNA molecule. So down here on kind of the right-hand lower portion of the figure, we see the DNA molecule kind of no longer in its double helix, but straightened out so that we can see the A to T and C to G complementary patterns. So those are all your nucleotides. The base pairing is attracted by hydrogen bonds, and then that molecule winds up into the double helix format. And that's kind of what we've already talked about. But now we're going to further condense and package this up so that it is protected while it's undergoing particularly division process in the nucleus. So we're going to wrap this DNA around specialized proteins. These are known as histones. And we can take 3 or 4 turns around a histone. That's actually known as a nucleosome. When the DNA is wound up like that, it cannot be used for transcription. So this is one way that we can kind of control gene activity. When the DNA is all tightly packaged up, the genes are not expressed. We're going to further kind of coil those nucleosomes up, and this coiled DNA is known as chromatin. All together, we call it a chromosome. So the gene is only that specific portion of the DNA that is going to be transcribed to messenger RNA and used for protein synthesis. So in all the cells in our body, like we said, the somatic cells, you would have 46 chromosomes, and they are in pairs. There's actually 23 pair because you got one copy of each of them from each parent. So in your somatic cells, like your skin or your organs, you would have 2 sets of chromosomes. And these are actually going to kind of be paired up with the same information about the same trait. In other words, you get a gene for hair color or eye color from your mom, and you get a gene for eye color from your dad. So these chromosomes that match up this way are known as homologous pairs. So they are going to contain the same set of genes for the same traits. So when we look at those genes, we see that they are on homologous chromosomes and they're at the same position. So in addition to our autosomes, we have 22 pairs of chromosomes that just define most of our inherited traits. But we also have a specific set known as our sex chromosomes, and this is going to determine our biological sex. If you get 2 X sex chromosomes, you are genetically female. If you get an X and a Y, you're genetically male. And under normal circumstances, the Y carries the information to produce the male phenotype. In other words, the male reproductive tract, the testes, testosterone production, secondary sex characteristics. So the things that we typically or phenotypically describe as male are initiated because of that Y chromosome. In females, the lack of a Y chromosome and the presence of those two X chromosomes yields to the development of the female reproductive tract and our female's sex characteristics in normal circumstances. So, in addition to producing somatic cells or sex cells, let's just look at what a typical cell goes through in its normal cell cycle. So think of your day that you have various patterns of activity throughout your day. Well, so this is true for your cells, especially dividing cells. So if you think about your skin cells, they're undergoing mitosis every day to produce new skin cells. And you're always shedding dead ones off the top surface of your skin. So they're always undergoing mitosis. And there's two major phases we want to look at. The interphase, which is kind of a growth and development, and then the actual cell division portion, which is mitosis. So mitosis is going to produce two identical daughter cells from one original body cell. And this is a duplication of basically the nucleus and the chromosomes that are in it. So here's a graphic of what's going on. So let's just look at the right-hand side. Following cell division, you're going to have two identical cells. Well, let's now take one of these cells through the next 24-hour process. After mitosis, it's going to enter this period known as G1, which is a growth phase. This is where the cell is going to increase in size and gain the ability to further undergo division. Before it can do that, it needs to duplicate its DNA. So this next section is known as the S phase, and this is where all the chromosomes are duplicated. So all 46 chromosomes will be copied through that process of DNA replication. Following the successful copying of the DNA, the chromosomes will consist of two sister chromatids. Those will be identical. We're going to have some more growth and additional multiplication of the organelles like the mitochondria until the cell is ready to undergo formal mitosis. Now notice up to this point, the G1, S, and G2 phases are in blue, and they are known as interphase. Cells normally spend most of their day in interphase. But when all the conditions are correct, they can enter the mitosis. And this is happening with the chromosomes in the nucleus. We begin with prophase to metaphase to anaphase and to telophase. At this point, we have our duplicated chromosomes have been separated, and then cytokinesis actually splits the cell into two new cells. And this just continues on about a 24-hour period for cells that are regularly dividing. Now, not all cells divide. For instance, your nerve cells typically do not reproduce themselves once they are established. And this is why nerve damage is such a problem. However, your skin, liver cells, certainly any of your epithelial linings like inside your gut, these are always and constantly undergoing mitosis. So we talked about interphase, which is basically the period for growth and preparation. That's the G1, S, and G2. During the G1, we have a period of major growth. During S phase, we're going to duplicate the DNA and have two sister chromatids which are genetically identical. And during G2, we have more growth. So then we're going to enter into mitosis, and we need to take all that spaghetti stringy DNA chromatin, which is in the nucleus, and begin to condense it down so that it's safe to take through mitosis. So the first thing we need to do, notice here we have that kind of classic X-shaped chromosome. That's because these two are identical copies held together in the center by a centromere. So we're going to wind all that up, package it around histones, wind it up again, and condense this chromosome into what you're probably used to looking at. Here we have two sister chromatids and a centromere kind of holding it together. And you can actually see these under the microscope. So above it is kind of looking at the condensed chromosomes. And when you see them in this form, remember, this is actually copied. There's actually two sets of the complete material here. So this would happen in the first phase. This is going to be during the first phase of mitosis. So let's take a look at that here. Over on the left-hand side, we see the chromosomes are not yet duplicated. During interphase, during that S phase, we're going to duplicate them. Then here they are duplicated, and we've actually lost the nuclear envelope. Notice how there's no more nucleus now. And we've started to form these spindle fibers, which can attach to the centromere and actually pull apart our sister chromatids. Notice how that allows us then to separate the cytoplasm through cytokinesis and create two new identical cells. So we talked a little bit about which cells divide and which cells do not. Well, when you were a single cell zygote, all of your cells are dividing because you need to grow into the embryo and then the fetus. So these cells are going to be rapidly dividing and also differentiating because some are going to become your nervous system, some will become your circulatory system, different organs, your skin. So not only do they divide, but they also differentiate. Now, when we get a little bit older, we have a lot of body cells involved in still growing. Not so much differentiation because now you have a heart, but it's going to get bigger. You have lungs, but they're going to get bigger. And we may also need to do repair. You know, when you skid your knee on your bicycle accident, you need to repair that skin surface. So that we use that mitosis for that. Now, when we become mature adults, some of the cells will lose their ability to divide. And like I mentioned, your nerve cells typically do not divide in the adult. Now, some typically don't divide, but they can. A number of your organs maintain a stock of stem cells. For instance, the liver. If you were to lose a large portion of your liver or have damage, basically they can recruit these stem cells to step in and begin dividing and repairing. Some actually do divide throughout your life, such as your skin cells and the lining of your digestive system. So let's look a little more closely at the process of mitosis. We're going to talk about prophase, metaphase, anaphase, telophase with cytokinesis. So during prophase, as we looked, the chromatin needs to condense with that DNA wrapping around the histones. We lose the nuclear membrane and we begin to form those spindle fibers, to reach out to the chromosomes. In the next phase, which is metaphase, we're going to have those mitotic spindle fibers reaching out and attaching to the chromosomes. The chromosomes are going to line up in the middle of the cell, and then they are going to be separated during anaphase. During telophase, we have the reforming of the nuclear envelope, the mitotic spindle apparatus dissembles, and then the chromosomes can actually decondense and go back to being available for gene expression. Here it is looking at a cartoon, but also a living cell. At the top is actually an embryo of a fish. So a fish egg in the process of dividing to become, a fish embryo in the process of dividing to become a fish. So if we look at interphase, notice there is a clear nucleus and the chromosome is not, or the DNA is not condensed. So at the top, you can see the nucleus, but you don't really see individual chromosomes. You just see the area where the chromatin is. As we enter into prophase, we notice the the chromatin is condensing and we're starting to see individual chromosomes. Notice also the centrioles producing the spindle fibers are moving to either pole to the edge of the cell. We then have all those chromosomes lining up in the center. This is metaphase. You can remember kind of middle phase. All of our duplicated chromosomes are lined up, and there are spindle fibers attached to them at the centromere. And if you look at the fish embryo, you see the dark blue representing the chromosomes and kind of the star-like fibers representing our spindle fibers. As the chromosomes begin to pull apart and be separated, we call this anaphase. And notice now we've separated each of the sister chromatids and we're pulling them towards the opposite poles. So in the fish embryo, you see the dark blue chromosomes beginning to separate. And in telophase, we have a much more pronounced separation, and we can even see the beginning of that cytokinesis where we're going to officially form two cells. So we have the new plate forming, which is going to separate one cell into two. And so at the end of cytokinesis, we have two new cells. The chromosomes have decondensed, and there's only one centriole. The nuclear envelope has reformed, and that'll be how the cell spends most of its life. So what is cytokinesis? This is specifically the division of the cytoplasm, and it's accomplished in kind of a really cool way. It's like a pinching off. There's a ring of microfilaments, and they contract around the midline of the cell to pinch it off into two cells. So here we have, for example, a cartoon of a cell that's undergone the telophase, but now we have to separate the cytoplasm. So we have microfilaments that come around the center of this cell, like around its waist, like a very tight belt, and begin to contract and actually pinch off the cytoplasm until the new membranes form and we have two separate cells. So looking at the chromosomes more closely, this is particularly important when you do have a developing embryo and you want to know, has everything gone well? Number one, is it a girl or a boy? But does it have the right number of chromosomes? Are they intact? Has there been any damage? So we can look at the chromosomes. And this is, in fact, a human karyotype where we've gone through and matched up the homologous pairs. So here's 2 of chromosome 1 One of those would have been from mom and one from dad. The same for chromosome 2, 3, 4, 5, 6, 7, 8, 9, 10. All of these are what we call our autosomes. And then we have the 23rd pair is our sex chromosomes. Could be two X's, could be an X and a Y. If there's been a problem, there could be different combinations here. So this would be a normal karyotype. Let me back up on that for a minute. If you were looking at the karyotype and you saw 3 copies of this number 21 gene, we would call that a trisomy. Trisomy 21 is also known as Down syndrome. So we want to look for irregular numbers. We also want to look to see if there's been any major damage. So you notice, you could imagine parts of these chromosomes could break off or even flip around, as happens in rare cases. So let's look at the process of meiosis. I'm actually going to do that in a separate video.